we'll start. Okay, the question is why archaea resist lysozyme and penicillin? In the bacteria, there are different type of the bacteria. Archaea is one of the most primitive class of the bacteria. And next, eubacteria. So these are the two different varieties of the bacteria. It is also called as archaebacteria. It is the most primitive one. And so the question is related to penicillin lysozyme. See, penicillin is an antibiotic. Already we discussed about it. Antibiotic. It acts on the cell wall of the bacteria. And lysozyme also acts on the cell wall of the bacteria. Usually it is one of the mechanisms used by the bacteriophage for the lysis of the bacterial cell wall. Okay. So these two, penicillin and lysozyme, they act on the cell wall of the bacteria. And here what they are asking, why these two antibiotics like penicillin and lysozyme, why they cannot act on the archaebacteria or archaea? That is the question. So here, if you see, so what are the archaea? So these are naturally resistant to penicillin lysozyme because their cell walls completely lack peptidoglycan. So that is the answer actually. What is peptidoglycan? It is the material composed both of NAM, that is N-acetyl muramic acid, another is NAG, that is N-acetyl guanosine. So here these two are important. They are cross-linked. They form the peptidoglycan. So NAM and NAG. These two are the components present in the peptidoglycan. But this peptidoglycan is absent in the archaea. Archaebacteria, they include, there are diverse variety of the bacteria which are present in archaea, like thermophilic bacteria, which grow in extremely high temperature. So those bacteria which belong to archaea, they grow in extreme conditions, like highly acidic conditions. Acidophilic bacteria, they are called acidophilic. They can grow in even highly acidic conditions. And in high temperature conditions, those are called a thermo. So they are all included under archaea. So they have cell wall, but in their cell wall there is no peptidoglycan, there is no muramic acid, there is no NAM, there is no NAG. So penicillin, actually how penicillin acts, it blocks the creation of the cross links in the peptidoglycan, because peptidoglycan is synthesized by the cross linking between NAM and NAG. So penicillin, it prevents the cross linking. So it prevents the formation of the peptidoglycan. Without peptidoglycan, bacteria cannot survive, because peptidoglycan is the component of the cell walls in the bacteria. And lysozyme also, it acts on the beta-1,4 glycosidic bonds in the peptidoglycan. Okay, so these two are the different mechanisms. Penicillin and lysozyme both act on the cell walls of the bacteria. How they act? Penicillin, it prevents the cross-linking of the peptidoglycan. It prevents the synthesis of the peptidoglycan. And lysozyme, it acts on the beta-1,4 glycosidic bonds which are present in the peptidoglycan. But archaea or archaebacteria, they lack peptidoglycan. There is no peptidoglycan. And also they have pseudomurine. That means instead of beta-1,4 glycosidic bonds in their cell walls, they have beta-1,3 glycosidic bonds. So because due to these reasons, these both penicillin and lysozyme cannot act on the cell wall of the archaea. They can act on the bacterial cell wall, but they cannot act on the archaea. So you should remember, because in future they may ask on different topics how lysozyme acts. It acts on the beta 1,4 glycosidic bonds present in the peptidoglycan. And penicillin prevents the cross-linking of the peptidoglycan. So you should remember these points. Okay, next. So here, what are the options given? Why archaea resist lysozyme and penicillin? Here they did not give that there is no peptidoglycan in the archaea. They gave that absence of muramic acid. Okay, NAM, N-acetyl muramic acid. Instead of NAM, they just gave that absence of muramic acid. Since there is no peptidoglycan, there is no muramic acid also. So that is the right option. And they gave different options like absence of proteins, absence of phospholipids, absence of glycoproteins. These are not the right options. The right option is absence of muramic acid or absence of the peptidoglycan. That is the right answer. Next, identify the correct sequence of events in bacteriophage replication. So you know what are the events which present in the bacteriophage replication. The bacteriophage, first it comes and binds to the cell wall of the bacteria. That is called as adsorption. And it releases the lysozyme, okay, to lyse the cell wall of the bacteria, and it enters, that is called entry. And next, the nucleic acid of the bacteriophage, that means the DNA, double-stranded DNA, it enters into the bacteria, that is called nucleic acid. And later, DNA synthesis or replication occurs. And next, assembly. Next, lysis. These are the sequence of the events which take place when the bacteriophage infects the bacteria and when the replication of the bacteriophage occurs. So, bacteriophage replication occurs. Bacteriophage is a virus. Its replication occurs within the bacteria. When the bacteriophage infects the bacteria, the replication occurs within the bacteria. So these are the sequence of the events. Once again, we will see what are the steps in lytic cycle. If you see here, host recognition. First, bacteriophage, it recognizes its host bacteria and it binds to the bacterial cell wall. Okay, and it has tail fibers. Bacteriophage has tail fibers. With the help of the tail fibers, it attaches to the bacterial cell wall. That is called as adsorption. Okay, and next, it releases the lysozyme and enzyme, and it creates a hole within the bacterial cell wall, and through that hole, the bacteriophage injects its DNA into the bacteria. That step is called DNA injection. Next, once the bacteriophage DNA enters into the bacteria, it replicates, it forms several copies, and based on this bacteriophage DNA, the bacteriophage heads, tails, and all the spare parts of the bacteriophage are synthesized. That is called as synthesis phase in the bacteria. And next, assembly of the bacteriophage parts, heads, tails, and all those parts of the bacteriophage, they assemble. That is called assembly stage. And finally, what happens? They release these bacteriophages. They release the enzyme lysozyme again. It breaks the cell wall, and all the bacteriophages are released from the bacteria. This is called as lysis. So all these are the different steps of the bacteriophage replication. It is also called as lytic cycle. Okay, so you have to remember all these sequences. So this is the right option: adsorption, entry, nucleic acid, and capsid synthesis, assembly, and lysis. So these are the right steps, sequence of the all these. Next, what is the correct sequence of zero arc? It is a part of succession. We see hydrosphere and xerosphere. Xerosphere is also called as zero arc. Okay. So what is the correct sequence? Again, they are asking about the sequence. What is the first step in the xerosphere? What is the next second one? What is the third one? What is the in a sequence? So here they give the pioneer stage, foliage, lichen stage, herbaceous stage, shrub, climax, forest. So these are the sequence of the steps given here. First option, and in the second option they give pioneer or crustose lichen stage, foliage lichen stage, moss stage, herbaceous stage, shrub stage, and climax forest. So it is much better than the first option, because in the first option moss stage is absent, moss stage is missing. Okay, so the second option has all the stages, and third option foliage lichen stage, here the crustose lichen stage is missing. First they gave the foliage lichen stage, next crustose lichen. So it is wrong option. And crustose lichen stage, foliage lichen stage, moss stage, shrub stage, climax stage. Here again the herbaceous stage is missing here. So all the stages in a right sequence, right order given in this one. Pioneer stage are called as crustose lichen stage. Next foliage lichen stage, moss stage, herbaceous stage. Once we will see this here, what are the stages are present? Stages of the xerosphere, it is also called as xeroarc. Pioneer crustose lichen stage, that is the one. Crustose lichen stage, this is called, because pioneers are called, they are called as crustose lichens are called as pioneers of the lithosphere, because they start their journey beginning from the rocks. They are able to start their life on the rocks. So they are called as pioneers. Next, after the crustose lichen stage, there is a foliage and fruticose lichen stage. And next, after the lichen stage, moss, that means bryophytes, funaria, polytrichum. These are called as moss. And after the moss stage, herb stage or herbaceous stage. After the herbs, shrub stage. And after that, tree stage. After that, climax stage. So here you see pioneer stage. It is started on the rocks, and there is no soil formation. In the evolution, you see at the beginning, the earth surface contains several rocks. Due to the activity of the several organisms, by chemical factors, by biological factors, the weathering of the rocks took place due to the growth of the plants in the rocks, on the surface of the rocks. So due to the several biological and chemical atmospheric factors, the weathering of the rocks occurred and soil is formed. Here, if you see lichens, crustose lichens, they started their life on the surface of the rocks. And second, foliage lichens, fruticose lichens, next mosses, herbs, shrubs, and trees. Finally, climax stage, forest. So here there is a much soil formation. What is the difference here? The soil is enriched by the growth of the plants. The soil is enriched due to the plants. Once the plants, after death of the plants, those plants are degraded, decomposed. It formed humus. So soil is more and more enriched, and finally it becomes fertile. That soil is fertile, and now finally that soil is suitable for the growth of the thick forest. So that is called as climax stage. Okay, so these stages in the hydrosphere and xerosphere you have to remember. Okay, next. Which of the following is a saprophytic food chain? So we have seen several types of the food chains in the ecology. So saprophytic food chain means the here, if you see it is also called as detritus food chain. Detritus, because when the leaves are fallen in the water, those leaves are decomposed by the activity of the several bacteria. So here that food chain is started by the dead leaves. So it is called as detritus food chain or saprophytic. What is saprophytic means? The organisms which depend on the dead organic matter. So it is either called as detritus food chain or saprophytic food chain. It is started by the dead organic matter. Usually leaves, by the dead leaves, decomposed leaves. These are eaten by insects like woodlouse, and they are finally eaten by birds on the trees. So this chain is called a detritus food chain or saprophytic food chain. So here, what they ask, which of the following is a saprophytic food chain? Oceanic food chain, pond food chain, terrestrial food chain, or detritus food chain? So the right answer is detritus food chain. It is started by the dead organic matter. Next, reduced scale-like leaves and modified leaves into spines are the adaptations shown by. So we know that this is also belong to ecology, adaptations of the plants, adaptations of the hydrophytes and xerophytes. Here they are asking about the reduction of the leaves, either in the form of the scale leaves or spines. It is related to xerophytes. Okay, it's a very easy answer. Hydrophytes is not hydrophytes, it is xerophytes, because to avoid the transpiration, xerophytes, okay, due to the scarcity of water, so they reduce the transpiration by modification of the leaves into either scales or spines. Okay, that is the adaptation of the xerophytes. Next, part B. Which spore stage is considered as sexual stage in rust fungi? So what is the rust fungi? Puccinia. Okay, Puccinia grami. So it causes rust disease particularly in wheat. So it has several types of the spores. What the options they give? Iridospore, teltospore, basidiospore, pycnospore. So these are the different spores formed in the life cycle of the Puccinia. We already discussed Puccinia, the primary host and secondary host for the Puccinia. Puccinia is a fungus. Okay, it belongs to the it belongs to the class Basidiomycetes or Basidiomycotina. So it has several stages in this life cycle. Once we'll see. See, this is the life cycle of the Puccinia. There are two plants involved. The primary host is wheat. The secondary host is barberry plant. Okay, it acts as a vector. So here, in the life cycle of the Puccinia, we see the basidiospores which are present in the soil. They infect the secondary host, that is a barberry plant, and it has pycnidium or spermagonium and ascidium or acium. So on the upper surface we see the pycnidium or spermagonium or spermatogonium, and at the lower side we see the ecium, okay, or eciospores. So from the pycnidium, pycniospores are released. From the ecium, eciospores or aciospores are released. And next stage is here, you see they infect the from the barberry plant, the ichthyospores, they infect the wheat stem and wheat leaves, and they form the iridium, also called as iridinosporangia. They produce iridinospores, which are red brick color. So, since due to the color of these spores in the form of the rust, it is also called a red rust. And next, they form teleal stage. Okay, and they produce teleospores. And those teleospores, here we see each teleospore has two nuclei. One is plus, another is minus. Okay, and they fuse together, and after the fusion, they produce 4 basidiospores after the meiosis. So this is the sexual stage. In the life cycle of the Puccinia, what is the sexual stage? The teleospores. After the formation of the teleospores, these two nuclei are fused and it forms diploid zygote. It undergoes meiosis and it forms 4 basidiospores. So here the question is, which spore stage is considered as sexual stage in rust fungi? So teletospore is considered as a sexual stage in the life cycle of the Puccinia. And one more question related to the Puccinia. What they ask, how many spore types appear in a macrocyclic rust life cycle? So they asked indirectly about the Puccinia life cycle. What is the macrocyclic life cycle? Which involves all the stages of the life cycle. That is called as macrocyclic. Microcyclic is if the life cycle involves only few stages, that is called macrocyclic. Macrocyclic means all the stages of the life cycle are involved. So how many stages are there totally? That is the question. 2, 3, 4, 5 they give. So here, if you see pycniospores, this is pycnidial stage, acidial stage, and iridial stage, and teleal stage, and next basidial stage. So we find 5 different types of the spores in the life cycle of the Puccinia. So the right answer is 5. How many spore types appear in a macrocyclic rust life cycle? The answer is 5. Next question. What is the terminator codon in mitochondrial DNA? We already discussed in the normal human DNA or in other DNA. There are termination codons present. Usually, we see actually in the RNA there is a uracil, but in the DNA there is a thymine instead of the uracil. So here in mitochondrial DNA, already we discussed that UAA, UGA, UAG are the 3 codons. These are called as nonsense codons or termination codons. But in mitochondrial DNA, what is the termination codon? That is the question they ask. That is the answer is AGA. Okay, AGA is the right answer. Next, which plasmid is used in genetic modification of crop plants? For example, BT cotton. To produce the BT cotton, which plasmid was used? PBR 322? No, it is mostly related to the cloning in the bacteria. This PBR 322 is very famous for the cloning in the bacteria. It is not related to crop plants. And F plasmid, fertility factor. It is related to the conjugation of the bacteria, the plasmid. So it is not related to the crop plants. And RI plasmid, So RI plasmid, it is a root inducing plasmid. It is particularly related to the secondary metabolite production. And TA plasmid, it is called as tumor inducing plasmid. These RI plasmid and TA plasmid, they are related to agrobacterium. In agrobacterium tumefaciens, TA plasmid is found. In agrobacterium rhizogenes, it is related to agrobacterium, but it is a different species. Agrobacterium rhizogenes. It contains the RI plasmid, that is root inducing plasmid. So the right answer here is the TA plasmid, because TA plasmid is very popular for transferring the genes into the crop plants. Okay, most of the times this TA plasmid is used to transfer the genes, to modify the genes into the crop plant. So that is the right answer. TA plasmid is the right answer. And one more question, which family has the floral formula? Here, see, you observe the floral formula here, BR, that means bracteate, BRL, that means bracteolate, and zygomorphic, percentage means zygomorphic, it is not symmetric, and bisexual here, and K5, C 1 plus 2 plus 2 within brackets, A within brackets 9 plus 1, and finally G1. So this one here, if you see, particularly if you see the corolla, you see K is related to calyx. There are 5 sepals, they are fused. That is called gamosepalous condition. Another is C 1 plus 2 plus 2 plus 2. This is the papilionaceous arrangement of the corolla. It is present in the family Fabaceae. Okay, and here A 9 plus 1 condition, diadelphous condition. 9 stamens are formed, they form a bundle, and there is a single separate stamen. That is called as 9 plus 1 condition. All these features are found in Fabaceae family. So now the options given, Asteraceae, it is not the right one. Anonaceae, it is wrong. Rutaceae is also wrong. Fabaceae is the right option. Okay, next. The gynoecium is bicarpellary and syncarpous in the family. So we can bicarpellary gynoecium and syncarpous. Where we find this condition? In Fabaceae. Fabaceae is monocarpous. There is a single carpel in the gynoecium in Fabaceae, monocarpal condition. And in Cisalpinaceae also, we see the monocarpellous condition. And next, Asteraceae, Mimosaceae. See, if you see the Fabaceae, Cisalpinaceae, Mimosaceae, all these have a single carpel. But Asteraceae has 2 carpels, that is bicarpellary and syncarpous condition. So the right answer is Asteraceae, bicarpellary condition, G2. It is called as G2 within brackets. Next, in which of the following genus heterostyle is present? Heterostyle, that means the style on the ovary at two or 3 different lengths. Distyle means the style in 2 different lengths. And tristyle, these are the 2 conditions. Heterostyle, in heterostyle we see distylic condition and tristylic condition based on the length of the style. So here, if you see, here we see on the top of the ovary, the style is longer, a long styled condition here. Longer with reference to stamens. Okay, here stamens are in lower stage, and if you see the style, it is very lengthy. And in the same members, in the same species, we see that style is very short. So we can see 2 morphological condition, bimorphic condition, okay, in the flowers. The flowers belong to same plant, same species, have two different morphological conditions, bimorphic flowers. In some of the flowers, we see the longer style, and in some of them short ones. So this is called as heterostyle. Here, it is also called as bistylic condition. It is present in primula. See, the right answer is Helianthus is the wrong answer. Primula, it is the right answer. And next, Chiropterophily type pollination is assisted by. Where we find Chiropterophily? The pollination mediated by bats is called as Chiropterophily. So the right answer is bats. We already discussed about the pollination, hydrophily. Okay, it involves the water. Anemophily, it involves the wind. Okay, so entomophily, it involves the insects as a pollinating agent. Ornithophily, it involves the birds. Birds act as the pollinating agent. So here the right answer is bats.